is my story. I was injured by Pfizer, batch number FA9093, on June 18th, 2021. Prior to my injection date, I had a severe, huge bruise on my right thigh. When I got the injection into my body, I came home 2 hours later and I started to feel a severe burning in my right thigh where I have the bruise. That pain was indescribable. I never felt pain like that before, especially from a bruise, especially after an injection. The last few days after that, I ended up getting crawling sensations, like there was bugs on my legs. There was vibrations, like internally. There was so much pain and throbbing and burning in my leg that just wouldn't go away. I thought I was clotting, so I kept putting my leg up on the back of the couch. For 2 years, I tried so many things. I've tried the NAC, the glutathione, the zinc, the quercetin, magnesium, vitamin D, fulvic acid. I've tried activated charcoal. I've tried it so many. Like last year, when I started detoxing and healing, I was on about 25 different supplements every single day. I wasted thousands of dollars on supplements. Then I started taking methylene blue a few months ago. Now the methylene blue started to help. Over the last 2 years, I've had about 50 symptoms or more. Most of it has been my nervous system and my circulation. I ended up figuring out that I have dysautonomia and POTS. I went to a neurologist and he told me that yes, I have dysautonomia and POTS, and that he's seen it after vaccines sometimes. And the only thing he told me to try was licorice root. Now, I never tried it, but after watching the conference with Healing with the Ages over the past weekend, they said to take licorice root. So the medical system know that there's venom in these shots. They know that licorice root, amongst other things, remove this venom from the body. So he knew. They all know. Like I said, I've tried so many supplements, so many detox protocols, so many healing modalities, and nothing helped me other than urine. Every day I felt like I was going to faint and pass out, or possibly just die randomly. I've had pins and needles, I've had electrical shocks, I've had muscle spasms and twitching and internal vibrations, burning, crawling sensations, throbbing and pulsating into my leg. I've had brain fog and memory loss, hives and random bruising. The list just goes on with how many symptoms I've had. And nobody's believed me ever since. I've literally had to heal myself. I've had to find different detox protocols and healing modalities through the vaccine injured community and the doctors with FLCCC and React 19 and other doctors online who are outspoken. Until I started watching the Jonathan Otto docuseries, Disease in Reverse. Something told me I had to watch this docuseries. And I had to plan it perfectly so I can keep notes and make sure that I had the information saved. So I watched the 9-part series, and on the first episode, when I first met Gloria, I knew that there was hope. I knew that there was some kind of healing modality out there that could be universal and help me plus other people. So for 6 months before the docuseries, I had this thought of drinking urine. But after the programming and the conditioning that we've been a part of since birth and the engineering, I was afraid. But then when I watched Gloria's testimony for being injured by Pfizer, healed by urine, I went upstairs and I started that day to drink my own urine to see what would happen. Instantly, I felt the internal problems slowly disappear. The head pressure, the tingling in my fingers and the throbbing in my leg, the burning in my feet, all these symptoms started to disappear. I was drinking urine for about 2 months until I decided to stop. I wanted to experiment. I wanted to see, because like I said, I was taking other supplements. I started the methylene blue, which really helped with oxygen supply and gasping for air. I was taking nattokinase and lion's mane along with the urine therapy, but nothing saved my life like urine. The other things were kind of like band-aids, but it was still there. I still felt the symptoms. I still felt the feeling that I was shutting down and that I could possibly give up at any moment because my thoughts weren't clear. I had suicidal thoughts. I felt like my body was shutting down. So like my brain was constantly fixated on the symptoms and the problems that was going on the inside of my body. But once I started the urine therapy, my life changed. So I was doing that for about 2 months. Then, like I said, I decided to quit the supplements, and I've been on a 1 week, no detox, no supplement, no healing modality protocol, just to see if my symptoms would come back. And instantly, the first day, the head pressure came back. Over the last week, I've had head pressure, my leg was throbbing and pulsating and a sharp pain. I've had burning in my feet again. I've had burning in my hands, pins and needles. I've had the internal vibrations. I had muscle twitching and electrical shock in my foot. I've had low energy. I've had depression, insomnia. I've had so many so far. God gave you everything you needed inside of your body. So I've decided to start back with the urine therapy. I'm going to make a video log every day. I will test it out for a week or two to see what disappears and what stays and how I feel. But I believe that the urine has saved my life. It is free. It's always available. Yeah, I want people to know that your urine is not waste. Your urine is not disgusting. God created your urine for a reason. Whatever comes out of your body, there's a purpose for it. Whatever is inside is a purpose, and your urine is ultra-filtered plasma from your blood. It has stem cells, vitamins, minerals, enzymes, anti-venom. It has so many components that it can literally heal almost any ailment in the body and reverse disease. So the doctors advise 2 things to get the venom out of the body from these shots, and that's nicotine. So if you're going to take nicotine gum, 2 milligrams every day. If you're going to take the patch, 7 milligrams every day for about 6 days. Then urine, you could do anytime. You could do it all the time. You could start off with any amount. You know, it's pretty wide open with what you could do with urine. If you want any more information, there is a website called urotherapyresearch.com with Dr. Group. He has a ton of resources that can help you. But there's nothing to be scared of. Your kidneys and your liver filter out all the toxins, and that's left in your body is your own medicine, and you can literally live off of your own urine for about 6 months if you had no water. So I just thought that I will make this testimony to explain that all these supplements, all these detox protocols, yes, it helped me minimize some symptoms. It helped me manage my injury with dysautonomia and POTS, but nothing helped quite like drinking my own urine. And I'm so grateful. I am so grateful that God was telling me to try the urine. And I'm so grateful that Jonathan Otto came into my life and I learned about disease in reverse and learned about Gloria's testimony. Because if it wasn't for her and if it wasn't for Jonathan Otto pumping out this information and not being afraid and helping the vaccine injured right now, I wouldn't be here today learning that I can fully heal from this bioweapon. It's been a blessing, and I'm so grateful. I'm really proud of this healing journey, which never stops. You're always going to be healing, you're always going to be growing, and always going to be learning for the rest of your life. But to know this has been a blessing in my life and my family's lives. So now my children won't find their mother dead in bed, won't find their mother struggling and in pain and suffering like I have been for 2 years. They now have a mom who knows how to cure herself from this venomous spike glycoprotein, which is from snakes and cone snails and their venoms. This shouldn't have to happen to humans, but at least there's a way out. So this is my first video to explain of my testimony on how I came down this route to the urine therapy healing modality. And I just hope that it reaches you near and far to help you with yourself or with a loved one. Just don't be afraid.
Hi, it's Jonathan Otto, and you're currently watching episode 2 of my brand new docuseries, Healing Genesis. Red-handed, exposing the deadly truth behind the global elite's deceptions. During the COVID pandemic, most of the information fed to us by the mainstream media and public authorities has been lies and deceptions. We've been told that natural medicine and giving the body the nutrients that it needs will not work, and that the deadly COVID jabs would be the one and only solution to the problem. We were also told to wear our masks, practice social distancing, and even stay away from friends and family who have not been vaccinated or refuse to get the jab. Public authorities have repeated time and again that COVID is not a man-made, but a naturally occurring virus that came out of Wuhan. But now we know the truth. Top experts and scientists have been slowly uncovering the truth, connecting the dots, and exposing the papers and studies proving that the entire COVID pandemic was nothing but a big lie. In this episode, we'll deep dive into the deceptions of the elites, the paper trails, and the research showing and explaining how the global elites have deceived us, spread fear, and furthered their agenda. Kicking this episode off is Dr. Brian Ardis, who will be shedding light on how venoms were used in the creation of COVID and the spike proteins. He reveals how these venoms are in our water supply, and how they've also been cunningly engineered and inserted into plasmids. Up next, we have Dr. Henry Ely, who will talk about some of the origins for the COVID pandemic. He brings attention to the Department of Defense's involvement and suggests a connection between vaccine manufacturers and bioweapons. Dr. Ely also shed light on the 1986 National Childhood Vaccine Injury Act, and he'll share why he believes this law actually protects companies that produce bioweapons, while at the same time putting a high price tag on babies. Following Dr. Ely is Dr. Lee Merritt, who shares why she believes that we're living in a world of deception. She also points out that COVID is not a sudden phenomenon, but is the result of a long-term plan with the intention of keeping the whole population sick or even killing us all prematurely. Dr. Merritt will also be exposing the harsh and heart-wrenching truths about what the government is actually doing, including human sacrifice, human trafficking, and even taking blood from our very own children.
I believe they're just using synthetic snake venoms and putting it in our water and creating COVID.
Yeah.
From that, what we have discovered is they didn't have to just put venoms in there. They actually used not even a viral vector. They used plasmids with the venoms inserted inside of it. The spike protein genes are venom genes, and they inserted them into plasmids that they could release in the air, water, food, injected into our food, and then they put it in the vaccines. And how it is they use plasmids, we have since learned, since Watch the Water, with the venom spike proteins, how they actually got the venom instruction into bacteria and yeast to replicate venoms in our bodies that they're calling COVID and long hauler COVID, that Carlo Brogna figured out the 36 different venoms. They were replicating in bacteria in the bowels of all the COVID patients, and they were ending up these venom proteins in the blood of everybody in that study. They used plasmids to instruct these bacteria and yeast cells to manufacture the venoms in our bodies to create the long hauler COVID symptoms. And now we've since learned, for sure, 100%, that they're putting those same plasmids with the venom spike protein in it in the COVID-19 mRNA shots. So people who have bacteria and yeast in their body, they are replicating the venoms inside their body after injecting the plasmids inside of them. Beyond that, what Ely, Group, Schmidt were all blown away with 4 weeks ago is I sent them a study. And in that study, it was discovered in 2003 that they took what are called plasmids, which are little circular DNA structures that look like a little hula hoop of DNA, and they insert the venom protein gene, called a spike protein gene, into the plasmids. In 2003, they took two DNA plasmids, and put pieces of the king cobra venom called cobra toxin. That was also found to be the spike protein of COVID. They took two pieces of the king cobra venom called cobra toxin, and they put a part of it inside of one plasmid. Then they took another DNA plasmid and put the other piece of the king cobra venom, cobra toxin, in that plasmid. So you had two halves of the king cobra venom's cobra toxin, that is the spike protein of COVID. They took those individually, they injected them into a mouse separately. They took this one and injected it into a mouse. Then they took this one and injected it into another mouse, and they waited. They wanted to see, would the mouse's own cells, which are mammal cells, would that mammal suck in the plasmid inside of its own cells and then spit out cobra toxin mRNA. In each of the scenarios with the half of the pieces of the king cobra venom in the plasmid that they injected into the animals, there was 0 side effects inside of those animals, and there was 0 confirmed cobra toxin mRNA manufactured in their bodies, in their cells. Then these scientists in 2003 took these two separate DNA plasmids with halves of the cobra toxin in each, and they decided to take both of them and inject them into mice. And they wanted to see, does the mouse's own cells start to manufacture together these two pieces of genetic material and splice them together in their cells to manufacture the complete cobra venom, cobra toxin protein. And then does it spit it out into the animal's body's tissues, intracellular fluid, and blood? And does it have the same effect as king cobra venom injected into an animal? When they separately injected both plasmids with the parts of the cobra toxin in them, the plasmids went inside the cells of the mammal, and the mouse's own cells took the two pieces of cobra toxin and did what's called trans-splicing. It actually put them together and made the complete, mature alpha cobra toxin mRNA, and then released it out of the cells. And every single animal died within 6 hours from the effects of the king cobra venom cobra toxin that was released into the interstitial fluid and blood of every one of these animals. 80% of every single animal that was injected with both separate plasmids, 80% of them died within 6 minutes. The mammal's own cells trans-spliced the two pieces of king cobra venom in two different plasmids, connected it together, and then spit it out. And it killed every one of the animals within 6 minutes, 80% of those, and then 100% of them died within 6 hours. They were able to confirm 20 years ago. Geneticists figured out they could create DNA plasmids with two pieces of king cobra venom, and that if they injected both into the animal, the own mammal's cells would manufacture the king cobra venom by trans-splicing it together, and then it kicks it out of the cell, and then the animals die. When I found this, it was the first confirmation for us that they were not only using DNA plasmids with venom-based spike proteins that were going to conjugate and be absorbed into bacteria called E. coli, which they have been confirming since 1999 they can do. They not only were doing that, and with yeast cells doing the same thing. They don't need bacteria and yeast to manufacture venom in a mammal. They figured out how to do what's called trans-splicing of two pieces of venoms into a mammal injected, and their body will make the full mature venom, and the animals will die within 6 hours, 100% of them. Remember, not a single one of the animals produced any of the king cobra venom mRNA from half of the cobra toxin injected to some of the animals with one plasmid, and then the other animals injected with the other plasmid. It took two injected plasmids to get their bodies to manufacture the cobra toxin mRNA for them. Why is this significant? When I sent this paper to Dr. Ed Group, Dr. Henry Ely, and Jana Schmidt, they almost fell out, because all of us up until this point, since Watch the Water, when we released that documentary, and Covenom 19-1, when we released that, we assumed that they had to use bacteria vectors and yeast vectors in our bodies to manufacture the venom proteins they're calling spike proteins. Nope, they don't need any bacteria or yeast to do it. If they inject two different plasmids with parts of the complete cobra venom protein, two plasmids into a mammal, the mammal cells will actually splice them together and spit out the entire venom protein, and the animal will react to the same venom protein as if the venom came from the creature into the animal in the wild, and they will die. Two things immediately happened with the other doctors I shared this with. Dr. Ed Group, Dr. Henry Ely, and Dr. Jana Schmidt. Immediately, Dr. Henry Ely texted to the group and said, "Oh my God, for the first time in history, a person to be considered completely vaccinated for the first time in history with these mRNA injections, it required 2 injections 2 weeks apart." So for the Pfizer and Moderna mRNA shots, you had one shot on day 1. 14 days later, you got the second one. Then and only then, you would be considered fully vaccinated against COVID. This is the first time in history a dual shot process was required to consider a person fully vaccinated. And Dr. Henry Ely's immediate response was, "Holy crap, does this explain why they had to do 2 mRNA injections to consider the individual vaccinated? Was it 2 different plasmids that had to be injected within a 2-week period with part of the spike protein venom in one plasmid and the other piece in the second shot?" 2 weeks later, and I immediately responded with this. 90% of all people who reported to VAERS had died after getting the COVID-19 mRNA shots. 90% of them died within 48 hours of the second shot, which fits the exact time frame for these mice in 2003 when they injected the two plasmids separately into the animal. Their body combined it, kicked out the full venom protein that killed the animal within 6 hours. For humans, we're much bigger than mice. It took 48 hours for 90% of them to die. Then Dr. Henry Ely went in to look and see, did they actually somehow differentiate the first Pfizer shot from the second Pfizer shot, and the first Moderna shot and the second Moderna shot for the people administering these mRNA injections. So, for example, we wanted to know, when a person came in for their first mRNA injection, is there a way that the person administering the shot to the individual would have been able to differentiate between the first shot that might have been given to the individual if they came in saying, actually, this is my second shot? Was there something these people would have been told to select a different Pfizer shot or Moderna shot for the second injection? We had no idea until a few weeks ago that Pfizer and Moderna had color capped a different color for the first injection for everybody to know this is the first one, and color capped a different color on top of the second injection. So when you, the innocent customer, showed up for your COVID-19 mRNA shot, Pfizer or Moderna, you would have been asked, is this your first or second COVID-19 shot? And you would have said, this is my first. They would have grabbed the gray color capped Pfizer shot, because it's the first one, and they would have injected you with that one. 2 weeks later, when you showed up to someone different to give you the second shot, they would have asked you this, is this your first or your second shot? And you would have said, this is my second. And they would have grabbed the purple capped Pfizer shot. This perfectly explains two different plasmids are being injected inside of these people's bodies, leading to the horrific outcomes we're seeing within 48 hours for the people who are dying from these shots. Nothing explains this better to why it is they had to differentiate the color caps to these mRNA injections other than to separate the fact that one's for the first shot, the other one's for the second. And my question for you is this. If they were identical, if all the shots were the same for the mRNA injections, why would you have to differentiate the first from the second? Why didn't you just have all batches go out with the same color cap? Because they're not the same. And they needed to make sure there was an easy way for the people giving the shots to differentiate between the first and the second. Now, the question for the audience should be this. How do you know there's plasmids in those shots called the COVID-19 mRNA shots? Well, there's 100% proof that's what's in these shots. Do you want to know how? Everybody at home, I want you to go and look up the NIH.gov's website. I don't care what search engine you use, type in NIH.gov and these words. How were the COVID-19 mRNA shots manufactured? And there's a leaflet, a little pamphlet, online pamphlet, that explains in perfect layman's terms how they manufactured the COVID-19 mRNA shots. It's right now on their website. Go look it up. It actually states in this pamphlet, for all audiences worldwide, you should know this. It actually says on the NIH's website right now, when we decided to create the COVID-19 vaccines, we did not put any SARS-CoV-2 virus in any of the mRNA injections. This is why we could confidently say, I'm quoting from the actual pamphlet, this is why we can tell you, you will not get COVID-19 from our shots, because we did not put the virus called SARS-CoV-2 in any of the mRNA shots. On the pamphlet, it tells you. My question for you is this. They told the entire world to take a shot called the Pfizer and Moderna mRNA, brand new gene therapy technology, greatest ever, never been used before in humans. They told you to go get this shot, the mRNA shots. That's what they pushed the hardest. In this document, it states, we did not put the SARS-CoV-2 virus in these shots. And all of you at home should be asking yourself, the 5 billion people that got these shots, why did I get the shot called mRNA injections if you were not giving us the SARS-CoV-2 virus for our body to make antibodies against for the future exposure, supposedly, to a SARS-CoV-2 variant. If you weren't being vaccinated against the SARS-CoV-2 virus for these COVID-19 shots, remember, they all told you, if you get these mRNA shots, you will not get COVID in the future. But COVID supposedly was being caused by symptoms being triggered by you being exposed to a virus called SARS-CoV-2 virus. If they didn't put SARS-CoV-2 virus in the shots, you should be asking yourself, what were they injecting inside of you to try to prevent? It's in the document. It actually states, we did not select the SARS-CoV-2 virus to put in these mRNA vaccines. Instead, it says, we selected the spike protein gene to be our vaccine candidate. And right next to this paragraph where it says that, it shows what they put in the COVID-19 vaccines. It shows a DNA plasmid, circular piece of DNA with the spike protein gene in it. And the next paragraph states, we synthetically manufactured the spike protein gene, which was found to be king cobra venom called cobra toxin, and bungarotoxin from the crate snake, and then segments of the rabies virus. But focus on these venoms. Why? You're going to learn a whole lot more in this documentary about the rabies virus segments and cobra toxin and the bungarotoxin venom proteins. They stated, we synthetically manufactured the spike protein gene, cobra toxin and bungarotoxin, and we inserted it into DNA plasmids. And that's what we put into the COVID-19 shots. In 2003, everyone, they figured out they didn't need bacteria or yeast. All they needed was to inject DNA plasmids into a mammal with 2 different pieces of the cobra toxin venom gene, called a spike protein gene, and they had to just inject 2 parts into the mammal. And 20 years ago, they figured out they can get the cells of the mammal to transplice the 2 plasmids together to complete and manufacture in our bodies, literally, transplice together the mature cobra toxin mRNA and release it into your cells. This is what they're doing with the COVID-19 mRNA injections. In fact, there is no mRNA inside those shots. They actually put DNA plasmids with the spike protein gene in them. And when you inject a mammal with the 2 DNA plasmids containing the spike protein genes, the body itself then manufactures the full spike protein gene mRNA in your body and release it. This is extremely troubling for every single human who received the mRNA injections. Why? You don't need E. coli bacteria or yeast in your body to manufacture this. You just need mammal cells to do it. And they have figured out decades ago how to make this happen and work 100% of the time. The speed at which the injuries are going to occur to anybody who received these 2 different plasmids called mRNA injections, the speed at which your symptoms are going to show up or disease outcomes are going to show up are going to be based on 2 things: your health and your microbiome. We're going to explain that. If you get these 2 plasmids injected into your body and you don't have any E. coli or yeast overgrowth in your body, your mammal cells are going to start manufacturing venom. It's just going to be slower than if you have E. coli or yeast in your body. Well, who are the people that have E. coli or yeast in their body? Right now, there's over 500 million people worldwide who have some type of diabetes. Diabetes is the inability to control blood sugar levels and maintain normal blood sugar levels. All diabetics have excess blood sugar. Diabetics are the ones most prone to have E. coli bacterial infections and candida or yeast overgrowth anywhere in their body, including in their bowels, in their kidneys, and in their bladder. You will often see this reoccur in diabetics called a urinary tract infection. It's usually caused by E. coli bacteria or yeast. The problem for you is you are the highest risk group for having now your own mammal cells manufacture the trans-spliced alpha cobra toxin mRNA in your body, in your own cells. But you not only have mammal cells that can engineer full venom proteins now, you also have E. coli bacteria eating on the blood sugar in excess in your body and replicating in your body. And then you have yeast in your body that's replicating by feeding on the sugar in your body. And those E. coli bacteria and those yeast, we know since 1999, have been engineered to suck in plasmids with venom spike protein genes in them and manufacture relentlessly and faster than mammal cells, manufacture the venom in your body. And this is exactly how they created the COVID-19 pandemic. We all suspected, I did too in the Watch the Water documentary, I suspected that they were putting the venom proteins that are synthetically manufactured around the world. I thought they were just putting it in our water systems and we were just drinking it. No. They didn't even need a viral vector. In fact, they didn't need a viral vector at all for this entire pandemic. They have been manufacturing DNA plasmids with the spike protein genes that are venom proteins in those plasmids, and they publish that they can release them in the air. They can drop them in the air. They can put them in your water systems, which they are. They also can inject it inside of our food. When you're hearing about Bill Gates and his Gavi group, Gavi, orchestrating farmlands around the world that he's buying up, and he is injecting avocados, tomatoes, and lettuce with the mRNA injections, they're creating mRNA injected food for us. When they are injecting the plants with these DNA plasmids they call mRNA injections, the DNA plasmids get sucked into bacteria and yeast. I don't know if you know this, but fungus and bacteria live in the soil of all your farmlands, and it gets drawn up into your plants. The plants need bacteria and fungus to grow. So in every single one of your vegetables, every single one of your fruits, they're injecting DNA plasmids in these mRNA injections, they're calling them. They're injecting them into the plants. And inside those plants, the E. coli bacteria and the yeast are going to start replicating venom proteins. And then you're going to eat them. And this is how they're going to continue to get these venom-laced plasmids into your body. What's the bigger problem? You all need to be paying attention to this. The CDC just reported a couple weeks ago there are outbreaks of E. coli bacteria on all spinach farms in America. Oh, really? Really? Why do you think they want you eating E. coli bacteria on your vegetables and on your plants? Because they need to get E. coli bacteria into you. Why? Those E. coli and yeast have been manufactured and engineered to replicate venoms as they do what's called conjugating. They suck in the plasmid, and that plasmid holds the instruction for the bacteria and yeast to manufacture venom proteins inside of you. This has been one of the biggest things we've realized. We're actually presented to the whole world. What it is they are utilizing in the form of plasmids in engineering bacteria, yeast, in our cells in what is called trans-splicing technology to get microorganisms in our body, in our own cells, to manufacture venoms, which will have a detrimental disease-causing, turbo cancer-creating side effect, which you're seeing worldwide from these shots.
Dr. Henry Elliott, we're talking about an intentional mass genocide. Am I using hyperbole?
You know, there's nothing more, Jono, that I would love to do than be wrong about what we have been researching. But it just keeps getting worse. And so there is no hyperbole, there is no sensationalism, there is no exaggeration necessary. This is horrific in every way that it could be genetically, and the implications for the very existence of life on this planet. So we don't need to sensationalize. That's how bad it really is.
Okay, because one of the things a lot of people, I think is that most people in the world took the shot, like, you know, the majority, well over, 70%, 90% in a lot of countries, almost like, 98% in some countries or whatever. And then a lot of people feel like, well, most people are okay. It's not as bad as what some people were warning and things like that. and sometimes I wonder as well, like you look down the streets and you see people walking around, cars driving, knowing that most people took this shot, and I wonder, like, what is going on here? Even though, on the other hand, I see the VAERS reports, the Vaccine Adverse Event Reporting System, and like how underreported that is, like a potential of up to as much as 100 times multiple, so 200 million reports potentially plus. So then, what do you think? Is it that did we dodge a bullet and that generally people are fine, or is there something set in motion here that people need to know how to switch off because what is coming is going to make what happened look like child's play?
Well, predicting the future is a fool's errand, I think, especially when we don't know all the variables that go into it. So I like to stay in the realm of what I can prove. What I can prove is, number one, that this is a military action. We know that the Department of Defense, the U.S. Department of Defense, is heavily involved and has ownership of it. If you just go back to the 1986 National Childhood Vaccine Injury Act, this is an injury act that protects the companies that produce these bioweapons, while at the same time putting a price tag on an infant, a baby's head, price tag of $250,000. If you inject your baby and your baby dies, which has happened to a lot of babies, one that comes to mind immediately is Holly Stavola. You can look that case up. What the government is going to do is argue with you that it wasn't the shots. And then if you somehow overcome that, they're going to award you a maximum settlement of $250,000. You have to think about the psychology of this. To create a law that protects a murderer of a child and pays a parent $250,000, that's akin to what we were doing in slavery, putting a price tag on a life. And for some insane reason, this is supposed to be about children, right? 1986. For some insane reason, the Department of Defense is mentioned throughout this law. And you have to start asking yourself, why? Why would the Department of Defense have any involvement in this? Why? Because the Department of Defense was actively working on bioweapons, and our research traces it back at least to 1968, but really it traces it back further. It traces it back to 1945, when General Patton and General Eisenhower were leading the Allied forces through Nazi-occupied Germany. And they get to extermination camps. Now, a lot of us have been taught, because they wanted to PC and dumb this down. You might remember Hogan's Heroes makes the Nazis into these lovable buffoons, right? They want to tone this down. So what the Nazis referred to these were as slave labor camps and extermination camps. They never referred to them as concentration camps. That's what Alan Dulles, the author and architect of Operation Paperclip, does when he brings 1,600 Nazi scientists from Nazi Germany, avoids Nuremberg, and plants them in the United States to continue their work on mind control. Well, you have to understand the mentality of a Nazi. The mentality of a Nazi is they expect to win the war, but they also are realists. They understand that their military is going to be decimated at the end of this war. And they're going to be in control of huge swaths of the globe with people living there that hate them. So there's going to be rebellions cropping up left and right. So what were the Nazis doing in these extermination camps to prepare for this? We don't have a military. We can't kill everybody, even though we would like to because we're psychopaths. What are we going to do? We're going to figure out how to control their minds. So what did the Nazis study in these extermination camps? Like Buchenwald and Auschwitz and Treblinka. These names we're all familiar with. They studied mind control. And what did they learn? The first tenet of mind control is nutrient deficiency. A person can withstand the assault of heavy metals on their neurology if they are nutrient deficient. So they made sure everybody was malnourished, number one. Then they started introducing aluminum. And aluminum starts improving suggestibility, especially with a little negative stimulus, and especially with a little visible negative stimulus that when you step out of line, you're going to get punished.
Is it true that it could make you like an antenna in some way to frequency?
Well, yes, ultimately, but I don't think they knew that at that time. I think they were just thinking, I want you to do what I want you to do, and I don't have the power to enforce it all over. I can't have rebellions cropping up. So what can I do to do this? I got to weaken you, put some heavy metals in there that completely dysregulate your nervous system, make you more prone to repeated messaging. So this is where you get the advent of the 24-hour news cycle right there. The Nazis were studying all this, impressionability. Now, Eisenhower and Patton show up in Buchenwald and they find 30,000 preserved organs that have been harvested, along with detailed scientific records of how they killed all those people. And what they learn is that the Nazis were studying two primary methods for mind control delivery of their bioweapons. Number one, vaccines. There's actually a study in Buchenwald where they were studying the typhus vaccine, and 700 of the 800 people that were administered the vaccine died. And the Nazis deemed that an unsuccessful test. Now, we might be thinking, well, oh yeah, because 700 or 800 died. Of course, that's unsuccessful. No, not to a Nazi. To a Nazi, it's unsuccessful because 800 out of 800 didn't die. You have to understand the psychopaths. So what we start getting into here is an understanding of really Sun Tzu in The Art of War. You have to think like your enemy. You have to understand your enemy. And the Nazis are crazy. I mean, we can get into the occult and we can get into Baphomet and all this other stuff that we've traced out, and the Gnostic Christians and all the symbology. We can get into all that because we've actually had to go down some of these ugly dark holes. I got to know my enemy. We get into this, and the Nazis settled on vaccines were going to be viable for delivery of whatever they wanted. But they had a second thing, and that second thing was water. What the Nazis were going to start doing after they won the world was building water treatment facilities in every major metropolitan area and ushering people from the countrysides into those areas so they can be concentrated there. A real concentration camp is a city. And now you can put whatever you want in the water, so if there's a rebellion cropping up in a city, wipe them out. You can wipe them out like that. I haven't studied whether Zyklon B is actually water soluble, but I think it is. And when you start to understand that, you go, wow, these psychopathic pseudoscientists are studying how to kill people and control them if they can't kill them. And Alan Dulles brings 1,600 of them here. You know what Patton and Eisenhower wanted to do to these murderers?
What?
He wanted to kill them on the spot. Both of them wanted to kill them on the spot. They were overruled. They were overruled because Alan Dulles was in with our president, Roosevelt, all right? And the decision was made, no. And then, of course, it becomes Truman. But it becomes, no, we're going to preserve that because they have information, and we don't want that information getting into the Russians' hands, right? This is right before the Cold War starts. So we bring those, I shouldn't say we, they bring those Nazis into our country and put them in the CIA and put them in NASA and give them unlimited funding and let them continue to hide in the dark and continue their work. And by 1953, you get MK Ultra, another project sponsored by Alan Dulles. But before that, guess what you get, Jono?
What?
You get, in 1948, the United States Federal Water Pollution Act. And guess what that does? It creates water treatment facilities and seizes control of all waterways. The government does.
Got it.
And guess by 1950 what starts getting put into the water of municipalities?
Aluminum, no, of course, fluoride.
You got it. And what's fluoride? What's sodium fluoride a waste product of?
What?
Aluminum production.
Okay.
So you're getting aluminum in. It's just by a different name.
Got it. Interesting. I didn't know that connection.
Crazy, right? And now you start fast forwarding, and by this 1960, almost every city, major city in the United States, which were growing exponentially, right? We were making fun of people who lived in the country, right? They're stupid. They don't know what they're, all this whole hit piece campaign, just like they did on cannabis, just like they've done everywhere. And when they want you to feel a certain way about something, they're great at masters of lying. And this is Joseph Goebbels, one of the number 2 Nazi. He was ruler for a day. He took his own life, that POS, right? That guy said, you tell a lie and you tell it big and you tell it often, and people will accept it as truth. And that's what they do. That's the playbook.
Yeah.
And that's what we saw with masks. That's what we saw with the damn shots. That's what we've seen with the whole vaccine program since it came in, started really growing in the 1960s. They got the first water treatment facilities established, and now they have their delivery system. And then they said, well, we're not going to get adults. Adult Americans are too free. They have actually too many nutrients in their body. We're not going to be able to control them. So who can we control? We can get the children. So what do they start doing? Tying the shots into going to school. And now you start getting the AMA buying in on it without questioning anything that's going on, not needing any significant research. Why? Because we'll get those doctors and we'll pay them to deliver these damn bioweapons into kids' bodies, and we'll make them think that they're doing a good thing when in reality what they're doing is committing attempted murder every time that they administer one of those damn shots.
And no actual vaccine safety studies, correct?
Zero. And especially none, Jono, and when it comes to how many shots, especially on the same day, in combination, zero safety studies on any of that. And I mean zero. And then you go look at the vaccine media and excipient summary, and what do you see? Aborted human fetal tissue. But I'm a witch doctor, an ND, right? I'm a witch doctor. But they're using aborted human fetal tissue. They're using aluminum. They're using mercury. They're using Darby kidney cells. They're using green monkey kidney cells. They're using fetal bovine serum. You know how they get that? They actually take a baby calf while it's still in the womb, still with its heart beating, and they draw and siphon the serum, the blood of that calf while it's in its mother's womb until it dies. And they use that to then put in the shots as a growth medium. Why? Because it's basically the purest blood you're going to get. Why? Because these people deal in blood. And you have to accept that when you're at war with somebody, especially as serious as these folks are. They deal in blood. Now, again, getting back to the main question, what can I prove? I can prove all of that.
Okay.
I can prove the vaccine schedule comes up. I can prove the water treatment. We all remember Flint, Michigan. Whatever happened to good old Flint, Michigan? Did they fix the water there? No. They had no intention of doing anything with that water, because water that comes out of your tap is the primary delivery system for their bioweapons. And right now, they're in the 3rd generation of their bioweapon development, and it's gone genetic. And that's where you have to be concerned, and you better respect this enemy, because they're not going away. We're going to have to put them away. We had a chance to put them away in 1945, and Alan Dulles said, no, we're going to keep this going. You don't take cancer and bring it into your body. That's a stupid thing to do. And that's what we did when we took those damn 1,600 Nazi scientists and brought them into this country, gave them U.S. taxpayer money, our money, and said, learn how to kill these useless eaters. At some point, we're going to need to get rid of them. At some point, we'll have our technology, our robots, and our AI, and this whole dystopian future that you see unfolding right now before our very eyes. We'll have it ready, and when that moment happens, we need to be able to push the go button. Well, what's the go button? The go button was COVID.
Dr. Lee Merritt, it's such a privilege to have some time with you in person. Like, I love so much getting the chance to connect with you personally. I love your insights, and I always enjoy the way you make me think about things differently, and that is super relevant when it comes to looking at world issues. Like, say, for example, why this is happening, why it follows certain patterns, how this plays out historically, and why that's important. That's a very interesting conversation, which we're going to get into. But also the whole concept of diagnosis and what disease actually is, and the smoke and mirrors game of this whole thing, and how they have us like intentionally injected with poisons and toxins that will create diseases that are known by names that make you think that they are like some kind of entity in and of themselves, versus just simply an exposure to a toxin, and then you're running after this impossible maze. And that's the whole thing really that we're helping people step out of. And I really appreciate you being here, Lee.
Well, thanks a lot. Thanks for having me. I always tell people I used to be a standard orthopedic and spinal surgeon, and now I'm a professional medical rebel. That's my website. Because you can't look at this and not realize we're being had. You know, what you call smoke and mirrors, I've come to believe now, is we're in a matrix. And it's a matrix of lies that are internally consistent. And they sound good. I think I'm working on a book called the Disease Delusion, because these things sound good, but in honesty, what they are is, as you just said, we have lots of labels. We went just not too long ago, we went from an ICD-9, that's the diagnostic code book that we use in medicine for the government medicine programs. We had 7,000, I think, diagnoses, or maybe 4,000, and now we have 70,000. So we make it sound in organized medicine that because we can label something, we really understand it. And one of my favorite examples is multiple sclerosis. You know, we claim that, and I'm a spine surgeon. For all my life, I did neurology evaluations. And we'd have somebody come in and they thought that it was their back or their neck, and I'd examine them and see them and think, no, that's not what's going on. Let's get, this may be something else, thinking about multiple sclerosis or MS. So I would get an MRI of the brain and the spinal cord, and it would come back with a reading and I'd look at it too, but it would come back as a reading from the radiologist. Oh, there are plaques in the brain and the spinal cord, and that means MS. So what is MS? They never take it the next step, and what causes it? Well, finally, we have a really good pathologist, Dr. McDonald, down, I think it's in Florida, who actually did very careful sectioning of the brain and spinal cord and found out that 100% of the dead MS patients he had autopsied had parasites in their brain and spinal cord. Some of it was actually grossly visible to the naked eye. So we just accepted, over my whole 45 years in medicine, we accepted that the smart people at the universities were paying attention to this stuff. And what we're learning, thanks to COVID, I will say, is that they weren't paying attention, that this can't be by accident. One of the things, too, I'll say is that after 45 years in medicine and seeing them telling us things that are completely wrong over and over and over again, you finally wake up and realize this is not by accident. This is a long-term calculated plan to keep us sick for some reason and to kill us prematurely. So that's my big picture on this, on what's going on. And I'm actually very hopeful, because I think once you recognize the matrix, you can step out of it. We have ways now, and we can understand we need to get back to root causes. Instead of, think about what we do. We spend so many billions of dollars every year in trying to make the precise diagnosis to go along with those 70,000 codes. But what if we took a step out of that whole idea and we said, wait a minute, what are the things that we know cause disease? Disease means dis-ease. It doesn't imply that there's a little infectious particle that goes out from me to you to make us sick. That's the lie that enables them to do what I call disease whack-a-mole, where they can just create a disease from thin air and get you afraid and then tell you to go out and get a vaccine. Well, if we understand that that's part of the matrix illusion, and that we actually only have a few really root causes of disease, that changes the whole paradigm. And I think that's where we are. I think we need to be paying attention to the actual root causes, and there aren't 70,000. Here's an example, one of the diagnostic codes is for getting burned while riding flaming water skis. Now, clearly, that's getting burned. The thing that caused the disease was fire. It wasn't the flaming water skis, and it wasn't separating it into different groups. That actually is that's a special code. It's like pecked by a chicken. That's also a code. And then pecked by a chicken subsequent event. That's a different disease, according to the ICD-10. This stuff is bizarre. But see what it does? It gets us focusing down in the weeds on minutia. That doesn't make any sense. So let's take, what if, and I believe this to be true, what if multiple sclerosis, acne rosacea, throat cancer, and rheumatoid arthritis, we think of those as 4 separate things. But guess what? They can all be treated successfully with antiparasitic medication. What's that tell you about those things? They're not 4 separate things. They're 4 different presentations of a common root cause. That's what we have to get down to.
Dr. Merritt, it appears that what's happening when it comes to diagnostic tests that are giving you all these different diagnoses of diseases, and then people are in these states of suffering, it appears to me that there is like an energy or an intention behind that thrives off not only death but suffering, and it has that intention. Obviously, the profiteering is the easiest way to explain these kinds of things, and everyone focuses there, and fair enough. Like, there's certainly money involved, but it's as if the suffering itself it's part of the outcome, irrespective of money. Do you think that's possible?
Oh, absolutely. It's like one big human sacrifice. Are we really such, we as people, we seem to just want to have our lives and raise our children and do everyday normal things. But it's like there's somebody in the sandbox that keeps stirring up stuff. And they create wars. I mean, provably now we know that the same people are funding both sides of most wars. So what's going on here? They're creating wars, and they're in the midst of the destruction, they make money. So, I mean, I'm looking at Ukraine right now, and it's a tragedy all around. But people have to realize that in the Ukraine, they're sacrificing their own people. Just recently, it came out over 500,000 young Ukrainian men have died. I mean, this is not a small number. And that's the death. Usually the disability is like a 7 to 1. So they've lost a lot of their workforce and a lot of people for what? Not only have some died, but the mothers can't find the bodies. They're saying, they're told by the army their son died, but they can't get the body back. And the reason is because they have what are called black transplantologists. Some of them from the United States are over there in underground, bunkers, parting out these guys. It's uglier than beyond belief, but their body parts are the #1 money in the world today. And they have been for a long time. We just haven't been paying attention. Wow. Body parts including blood products and including your liver, your heart, etc. And so these guys aren't just dying. These guys are, and anybody that knows about transplantation knows they can't be completely dead when they do this. So they're taking these young, injured guys and they're taking their organs. If you want to really understand the basis of the world, that seems to be it. Create misery, create fear, and profit from it. And the question you have to ask is, who's really behind all this?
That is really horrifying. Who is behind this?
Yeah, it is horrifying. And that's been happening to children. The Sound of Freedom, I thought, was an excellent primer for people that hadn't gotten their toes wet into the real issue of human trafficking. But it doesn't tell you the whole story. They stop short of telling you what really happens. And what really happens is after these children are used up in the sex trafficking, they're parted out for their organs. So, and adrenochrome, that's real.
Both Tim Ballard and Jim Caviezel are speaking publicly about that aspect as well, with the blood and adrenochrome. They're actually, yeah. I was surprised to hear Jim Caviezel talk about the enterprise of the blood of children.
Yeah, and actually, it's the blood of everybody. It turns out our blood supply, I remember my mother, she was a nurse, and all her life she volunteered for the Red Cross to give blood. And truly, we need some blood in our system to help trauma victims, for example, and other people. But it turns out the system was also for big profit. And you see this all around the world in various different things, where there's this apparently nice organization is actually making huge profits on human body parts. In fact, here's an example. Years ago, during the time that I think Clinton was maybe the lieutenant governor in Arkansas, and they used the Red Cross for this. They decided to start harvesting blood and plasma and things from their penitentiary system. And that's not necessarily the best group of people to harvest from, safety-wise. But in any case, they sent this blood, they sold this blood around the world, specifically to Canada. And Canada then had an outbreak of hepatitis C and AIDS, and complained, and the whole thing got shut down. But it didn't really get shut down, because what happened, there's some types of blood that are rare. And it turns out that Native Americans have some of that blood. And as soon as they shut down the official channels for this, some of they had an uptick in the disappearance of Native Americans. We've just not been paying attention. This has been happening for a very long time.
The hard part about this, Dr. Merritt, is that the information is so startling and it seems so impossible now of this horrific world that we live in that, I mean, people don't really want to hear about it because they feel like, well, what can I do about this? And it just makes me feel afraid to even live and be in public and, I feel like I'm going to get abducted out of my home. How do you help people navigate that?
Right. I mean, my friend said it, a sufficiently advanced intelligence is indistinguishable from madness. It does sound crazy, and the people talking like me sound crazy for saying some of this. But once you've looked into this for years, and you have enough data points behind you, it's like a murder mystery. I feel more like Angela Lansbury in Murder, She Wrote than I do about a physician doing scientific research these days. But the whole system is made to confuse us. Confusion is, there are two things that the enemy really is expert at, and one is making people fearful, because fearful people make bad mistakes, and the other thing is to totally confuse what's going on. And I will just say, I gave a talk actually at Red Pill on this year on it's time to name the enemy. And no matter how I look at this is not something new. When you look at whatever, I mean, pick the New World Order, that we hear Klaus Schwab bloviating about. The New World Order. Well, before that was the New Order of the Ages. That's what, Novo Ordo Seclorum on our bill, Yeah. But before that, it was called the Great Plan. This is a plan, just like the Masonic images, the Masonic symbols and rituals. This goes back to the time of the pharaohs. And this is provable. So a lot of this we are dealing with an ancient war against us, humanity. And we can argue, I call it the Babylonian Mafia, because I can trace through bloodlines, through genealogy, through DNA, through history and archaeology. You can trace it all to a pretty one area. And it seems to be that we're dealing with a cabal, or like I say, the Babylonian Mafia of ancient bloodlines that it's hard for us to wrap our head around, because we think all people are the same. But let's just look at movies and TV and the predictive programming in these shows. What are they always telling us? Think about Westworld is the predominant one, but whether it's The Matrix or Westworld or The Truman Show, there are a ton of, it's all about this. They keep saying over and over, there are people that look like us, walk like us, talk like us, but they're not us. And I can't escape that. I really believe that's true. It's hard. We have been so sold on the idea that everybody's the same. We all have the same motives, but it's just provably not true. For example, in psychiatry, and we've known this for a long time, when I was in medical school in the 1970s, they claimed that 4% of people were psychopaths. Now we're up to about 10%, they say. Now, so what is a psychopath? Well, think about, if we believe, as I do, that the COVID vaccine and all this going on is really a depopulation plan, then you've got a group of people out there that are intent on mass murder. That's what we call psychopathology. And what's the biggest diagnostic characteristic of psychopathology? Having no human empathy. In other words, not being able to understand why somebody, we look at people. We're as a human being, as a real human being, child of God, we look at each other and we can recognize each other by how our facial expression is, how we emote. We get signals, and we feel bad for somebody else. But psychopaths don't. They have no human empathy. And the way they learn to fit into society as small children, they learn to imitate human emotions. So what does that sound like? I mean, like the little kid goes to a funeral and he sees everybody else crying, and he realizes that he's going to look out of place if he doesn't cry. So he learns to pretend these human emotions, and they're very good at it. So we're dealing with a group of people here that are psychopaths, wherever they came from. I believe ancient Babylon and probably Sumer, but they're psychopaths, what we call psychopaths, because they don't really click with humans. They don't click with the rest of us. And yeah, it's kind of a creepy thing. The other thing is we're purposely being misled about psychopathology in the TV shows and the movies, where they have the FBI guy always showing you, like Jeffrey Dahmer, that they're lone, kind of crazy, creepy people. That's not what most psychopaths are. Most psychopaths are smart, well-educated, top of their game people. They can cooperate with each other when it suits them to further their interests. They can recognize each other, but we can't recognize them for the most part. And an example of that, when I was in the Navy, and I was a Navy surgeon and physician for 10 years. In the Navy, I did research a couple summers at NUMI, I think it was the Navy Undersea Medical Institute. And one of the things, there was a psychologist that was there, a PhD guy from Yale, and he was looking at different aspects of psychology of submarine existence, right? How you choose sonar operators, for example. But one of the things they looked at was how to prevent psychopaths from shipping out. Because you're going to be under the polar ice cap for 3 months. You don't want somebody that's a psychopath on board the ship. And that's the one psychiatric group they couldn't screen out, because they can't recognize them. They are completely hidden in our society.
Up next, we have Dr. Seema Nanda, who provides an in-depth explanation of the mRNA vaccine technology, focusing on its lipid nanoparticle delivery system. She likens the engineered system to a Trojan horse, which shields the mRNA from the body's defenses, allowing it to enter cells and initiate a targeted immune response. She mentions how Dr. Carrico and Dr. Wiseman's work in 2005 aimed to strengthen the mRNA's stability within the body by encapsulating it in lipid nanoparticles, and hints at how the COVID pandemic is definitely something that is not new, but was something that was strategically planned since in the early years.
Carrico and Wiseman, Dr. Carrico and Wiseman. So in 2005, they were trying to figure out, when they were thinking about mRNA as instructions for vaccines, they were trying to figure out, how do we make it so it doesn't break down in the body? Because the minute it comes into the body, it got shredded by the body, because the body goes, this is not us, this is fake, we know it, die already. So they tried to figure out, what can we do, what can we modify or tweak in the mRNA to make it stronger so it can bypass the system and hide in the body. One thing they did was they put it in a soap bubble, like a lipid nanoparticle. It's this big fat emulsifier. And it's kind of like a Trojan horse. Do you remember the story of the Trojan horse? They built this beautiful horse out of wood. So they give him as a gift, and they go, oh, what a beautiful gift. And then once they open the gates and let the horse in, then all of a sudden, inside the wooden horse, all these soldiers came out and, plundered and did all their bad things. That's what it's doing. It's in this bubble, this protective bubble, this lipid fat, which is lipid. Nano means the billionth, so it's very, very tiny lipid nanoparticles. So it's super tiny, and it's encapsulating this mRNA so it won't be broken down by the body. Very, very clever. Very evil, but very clever. So another clever thing that was done with mRNA is in order to prevent it from being broken down. In other words, degrade into the body, because again, the body is very clever. God made us very brilliantly, and he knows when something shouldn't be there. So these two scientists decide that they want to create this mRNA technology vaccine, and other things. They want to inject it into our foods. They want to inject into it so we can change the structure and the proteins that are in us. But they realized that we have to outsmart the body. And they tried many different things. And like I said, in 2005, they came up with this way. And in 2011, they produced that article that showed what they were doing with this mRNA. So how did they do it? If we go back to DNA and we understand that it's the molecule of DNA, it's considered a nucleotide, and those things base together, they pair together. And adenine pairs with thymine, AT, and guanine pairs with cytosine, GC. So one's a pyrimidine and one's a purine, and they go into a pattern And that's the double-stranded helix molecule. It's just brilliant. And it stays tightened up. It's woundly like a coil. And it does not open up until it needs to be replicated or made into a copy. So it stays together. Now, how you know it's a DNA versus an RNA is when the replication occurs. So again, it's wildly tied up, and then it needs to make a copy, and when it makes a copy, it opens up. So now the rungs of the ladder are opened up for the copy. So instead of being AT, adenine thymine, in DNA, it becomes AU, adenine uracil. And this is how we know it is RNA. So it's not in the nucleus, it's being replicated, so that way we don't mix things up. And again, another brilliant thought. So we have adenine and uracil. Now, they must have been testing all these different things. But they figured out, like I said, it's a molecule, a ribose molecule, which is a 5-carbon sugar. This is ribose, the molecule here. It's the 5-carbon sugar, and it's attached here. And this right here is called a purine, Purine, and it is attached via a nitrogen carbon. So this is carbon, this is nitrogen. Now, this is easily broken down. What they decided to do is they flipped it or rotated it 180 degrees. And you can see this here, it's rotated. So instead of being bound to the nitrogen, it's now being bound to another carbon. So when they flipped it, it made it stronger and not degradable. So this is the fake or pseudo uracil. And since it's attached to the ribose and everything, and it's called a nucleoside, they call it uridine. So it's pseudo-uridine. And this is kind of what they did in order to make it stable and being broken down and things like that. So that is brilliantly scary. And that's the best way to describe it. They figured that out, and when they figured it out, they said, oh, eureka, we found it. And that's how, in certain individuals, they do not have that effect, which is also quite brilliant. And I don't mean that in a good way. That is a very sarcastic way of saying brilliant, because if it was for good intentions, and let's just assume they were just trying to create this thing, help people out, whatever their reasoning is. But what's happening is it's not breaking down at all. So when you're injecting this into the person, it's causing them to continue to make the spike protein, which is mounting even a further attack on the body, and the patient can't recover. So, and what I mean by they can't recover, it's like I was saying to you before, it's too many cells, too much production, and then either their body shuts down, or what happens, and this is kind of more important, and I'm going to relate it back to the eyes, they have these docking. So once the spike protein is produced by the mRNA, because it's in the instructions, it docks into the endothelium, which means the inside of like a pipe. So if you think of a pipe is the blood vessel wall. So it docks on there. There's two subunits, subunit 1 and 2, and they bind to what we call ACE, ACE, angiotensin converting enzyme, ACE2 receptors. This is one theory, okay? So when it binds to that and it sits there, and then the other spike protein binds and the other spike protein. So now all of a sudden you're filling up a very narrow channel, or the blood vessel. And when you keep filling it up and all of a sudden it gets closer and closer and closer, what do you have? You have a closing of that channel, a bottleneck, a traffic jam, a clot. So it's not theoretically a clot that you have where you take an aspirin and it breaks down the clot. It's a filling up of all these red blood cells and everything, these proteins and everything, and it doesn't allow the passage, and that's why these clots are happening.
Concluding this episode is Dr. Andrew Kaufman, who will be discussing and helping us understand the controversies surrounding germ theory and contagion. Dr. Kaufman draws attention to historical experiments, including those conducted during the Spanish flu pandemic, which failed to show the transmission of diseases from sick individuals to healthy ones. He'll also explain the importance of conducting rigorous experiments to determine the true causes of illness and questions the role of viruses in causing diseases. It's Jonathan Otto here, and I'm with Dr. Andy Kaufman, and Dr. Kaufman and I go back actually, and we were just reminiscing before the cameras were rolling on our connection to the late Dr. Rashid Buttar, who was a dear friend of both of ours and somebody that we both mutually respect, and we feel, I feel Andy feels a commitment to sharing his legacy and continuing this work that he did so powerfully. And that's how we met. We met at a conference, advanced medicine conference, and I was in Australia at the time, but I was interviewing remotely, but we connected, and I've really respected and appreciated Dr. Kaufman's work. And I think that there are some others around the world, and Dr. Thomas Cowan, that are really seeing something about the myth of how we've been lied to about disease and how disease is transmitted, what the mechanism is. And it really does point people toward, one, what is the real toxic exposure that you can actually measure versus things that are so ominous and impossible to trace, but the actual toxic exposure, and this is my words, not yours, Dr. Kaufman, but and then in light of understanding the true toxic triggers, then look at how to help the body to heal and regenerate itself, and that to trust your body's self-healing mechanism and not to be afraid of things like viruses, I think is a really incredible and awesome takeaway. But that all to say, Dr. Kaufman, really respect and appreciate your work. You're somebody that's certainly leading in a train of thought that I think is going to continue to influence the world for many decades and centuries to come if we last that long as a civilization. So thank you for being here.
Well, that's very kind of you, Jonathan. And, based on your explanation, I'm not sure if I'm really needed. I think you have an excellent grasp on, the major points to take away. And, like you said, to help support the body's self-healing mechanisms. And so this is really, a mechanism, a message of empowerment, because we each can take control of our health. Once we realize certain truths, and we're not so, dependent on some kind of magic bullet to help restore us to a healthy state.
Yeah, no, that's fantastic. And so, I mean, what was this? Okay, for you, firstly, your background. If you could share your medical and your clinical background, that'd be great.
Sure. I studied biology as an undergrad at MIT, and I did various types of research there, both in the laboratories on campus and then also in a couple of different biotech companies. And so I got to see a lot of research that is about drug discovery and drug development and therapeutics and diagnostics from that perspective. Then I worked in the health department in New York City, in a CDC-sponsored position to do surveillance of AIDS cases. So I was very much involved in, reporting AIDS cases, reviewing records, understanding everything about that condition, although I didn't know the full truth about viruses back then. Then I went to school to become a physician assistant, where I started getting clinical experience in cancer medicine and hematology, and went back to medical school to eventually specialize in psychiatry, where I trained at Duke, and then forensic psychiatry, where I trained at Upstate Medical University, and I practiced and was held a teaching position at Upstate Medical University for several years before I learned enough to decide to make a break from the mainstream medical system and disclose the truth about healthcare, or what I know about it, to the public, and to study natural healing and try to teach both individuals and other health professionals about those principles, which just get amazing results, nothing like the mainstream health system.
So then, how does that relate to COVID? So now we have this huge thing that happens in the world in 2020, and yeah, start of 2020, and that this is now, transmitting. And now there's these multiple theories now that come out. For example, one is, we won't get into the later ones yet, but just the mainstream ones is simply that is the bats at the meat market, and then there's then even all these people are talking then about a lab leak, and then even the FBI now are saying, the lab leak is plausible, and that's probably the method and so forth. But then I'm saying that doesn't make much sense to me about like this just kind of escaping out of a lab, going to one person, then goes to the next person, the next person. And it really absolves any government or any institution or any confederacy of anyone that could have been actually putting poisons in our environment, for example. Let's not even discussing what those exact poisons were, but it's such a good way to absolve yourself of guilt. I'm not saying that therefore I believe that, but these were some of the big cases there that were then coming out. And because people are so indoctrinated on the concept of a virus being transmitted in something basically very much like a zombie apocalypse, where all you have to do is get bitten and then it just transmits, and then that person now and everyone bites each other, and then or some ethereal thing where it's breathed on somebody, then they get it, and then they get it, and then everyone becomes a threat then, and people that your family that you normally love, they're all like the enemy now, and this was such a great way to divide people. But what do you think of this concept and how that came out, and how what your theory relates to COVID and even the vaccines, but even the proliferation of COVID itself? Is this relevant?
Well, absolutely. And, the reason why you hear this word nowadays is because of COVID, because COVID, represents germ theory, right? Which is the theory that these invisible microorganisms, they're not invisible. You could see them in a microscope, but to the naked eye, they're invisible. That they are the cause of these diseases that are said to spread from person to person, like you described. And of course, there were a lot of scary things that happened during this time, and so without explanation. And this was provided as the one explanation for everything, right? And whether it occurred from some natural, accident, right, like from monkeys to humans or something like that, or whether it occurred by man-made accident, like the lab leak hypothesis, right? It still provided the explanation for all the phenomena that we witnessed, including, governments taking away our freedoms. So, of course, as you describe, all of it is based on the ability, right, of a illness to be passed from one individual to another, right, from person to person spread. And you mentioned several ways, right, that through the breath or other things. And this is, of course, we know called contagion, and we've all been told since we were very, very young tykes that all these illnesses, acute illnesses, work by contagion, right? We're not told that every illness works this way, right? Things like diabetes, for example, or heart attacks are not contagious, but all these acute illnesses that are blamed on microorganisms all have this contagion phenomenon. So here's the problem is that whenever in every situation where a scientist has tried to test contagion experimentally, right, where they do an experiment where they have people with the illness and then they take fluids or expose those people to healthy people, and they do this with animals too, but they've done many experiments in humans, they just don't make anyone sick. So this idea that there exists something in the world that causes an illness and can be passed from person to person simply is not true. There's only the belief of that, but every single time that there's been an experiment to try and show it, the experiment has failed. So some of the most famous experiments that tried to test this were done during the Spanish flu, where they had, it would be just like going into the COVID ward of the hospital. They went into the Spanish flu ward of the hospital and took people there that were, in the process of being very sick, and took all their different secretions, their snot, their saliva, etc. And they gave it, several different ways to groups of healthy people, and none of the healthy people got sick with the Spanish flu. And this was done in many other illnesses, the common cold, sexually transmitted diseases, and others in animals as well, even things like polio. And in all cases, it's been shown not to occur. So just by that research alone, we can determine that there couldn't have been a lab leak, and that even if there was a virus that existed, it couldn't have spread person to person, because that's just not how things work in nature. Now, I know it seems kind of difficult to believe that, because many of these experiments were done decades ago. And all of our lives, right, we've been told that this is the case. But the truth is, we never looked for the evidence. And we do observe patterns in our own experience that sometimes seem like one person is getting another person sick. But in truth, there's another explanation for that. And, there are many others that we're familiar with also, right? Like, we could both be exposed to something that made us sick, and whether that's a germ or not, that's a separate question, because we're talking about person-to-person spread of disease, and that's never been demonstrated. Now, we can certainly take the second part of that, which is, do these germs cause the disease? And it would be a very simple experiment to do, right? Because you just have to take the germ, get it in a pure form where it's just the germ by itself, right? And then put it in a healthy person and see if it causes the disease. But with many bacteria that are said to cause disease, right, before we even get to viruses, and things like cholera, for example, and yellow fever, when those bacteria have been taken by the natural route, like swallowed by healthy individuals, they did not get cholera or yellow fever. Okay, now there's other information about bacteria, but let's just go to viruses for a second, because that's kind of what we're really wanted to talk about. So these are said to be particles that are so small they can't even be seen with a regular microscope. They can only be seen with an electron microscope, which can see things that are 50 to 150 nanometers, which are billionths of a meter. And when they've looked for those directly in sick people, they're not able to find it. So they really wanted to push for viruses to really be the cause. So they created an experiment where they create the appearance of viruses being present. But what they do is they just take some sample from a sick patient, which is a mixture of things, right? So it could be like, for example, let's say they have pneumonia and they're coughing up sputum, right? That kind of thick, mucousy stuff. So what they do is they just take that, they run it through a coarse filter to remove some bigger debris, but who knows what's in that, right? It's whatever is in it is your body trying to get rid of stuff. And they use that to say that the virus is in there somewhere, right? But they never prove it. Like, they don't show that the virus is there directly by any means. Like, they don't take microscope images of that stuff from your lungs. They just put it in the experiment into the cell culture with the assumption there's a virus in there without any proof. And then they add some other chemicals to the cell culture. And by the way, the cell culture is foreign cells like that you could just buy at a commercial company, and they're usually monkey cells, but they could be even human, and they're usually modified in some way genetically. They could even be cancer cells sometimes. Okay, in one of the COVID experiments, they actually used lung cancer cells. And then they show that as the cells grow over several days, they sometimes, and they don't tell you how many times, by the way, like they don't tell you how many cultures they run and how many of the cultures get damaged. But it's a small portion. It's not all of them. It's not 25% or 50%. Okay, it's like somewhere less than 10% of the actual cultures they run show any damage. And when they do see that damage, right, and all they're showing is damaged cells in a culture, they take that to mean that there's a virus in the experiment somewhere from the lung fluid. But the thing is, they've done this experiment without even using the lung fluid, with just using the chemicals in a cell culture, and they still get cell damage. When they do this for experiment for a virus, they don't do that control experiment, which violates the scientific method. So somehow, since 1954, when the first time this experiment was published, and by the way, that time it was in a measles experiment, and it wasn't even given as proof of the existence of measles. But subsequently, shortly after, in other published papers where they carried out the same experiment, they said that this is proof of a virus in the sick patient, and then that has been carried forward since that time, that every published paper claiming to detect or discover a virus is done by that exact method.
This is the same reason why there's no double-blind placebos for vaccinations, because the information is just too damning, and so they do exist probably, but they're just not disclosed, and they don't want to know this information.
It's even worse for vaccine studies, because in order to get a vaccine approved, you don't have to do an experiment where you show that it actually prevents the disease. So let's say, you come up with a new measles vaccine. You don't have to do a study where you give 1,000 kids the measles vaccine and 1,000 kids don't get it, and then you wait until they're 10 years old and record how many cases of measles there were in both groups. You don't have to do that at all. All you have to do is give the experimental shot and then do a blood test some days later and show that there is a supposed antibody to measles. Of course, these antibodies have never been shown to predict whether there's a future case of measles or not. But that's the bar. So not only do you do not do a placebo-controlled trial, you actually compare it to the measles vaccine that's already on the market. You don't have to compare it to a placebo. And it's similar with chemotherapy drugs are also tested this way. You can't find a placebo-controlled trial of chemotherapy anywhere in the literature. If you did, no one would buy chemotherapy anymore, because they'd see that people lived longer without any treatment. There is an unwritten law in medicine about cancer that says, if you get chemotherapy, you'll live longer. But the thing is that there's just no evidence to support that. The evidence is to the contrary, but they bury it by never, any time that you look at any study and you see the absence of a control group, right? Because we were talking about with a placebo in a drug trial, that's a control group, right? That you give them a pill or you give them an IV, but it doesn't have the real drug that you're testing in there, because it's possible that just giving them the IV could actually give them a benefit. In fact, there was a major study done by the Veterans Administration in the United States where they were testing bypass surgery, cardiac bypass surgery. And they did sham surgery in half of the patients, where they just made a skin incision in their chest, but did not cut open their breastplate, the sternum. But the next day they didn't know, right? Because they just had a wound on their chest that was healing and painful. And it turns out that the patients who got the sham surgery did as well as the ones who got the bypass surgery. So you cannot ethically do a study, or you cannot scientifically do any study without a control group and know that the effect that you see is actually the effect in reality. Right? Because that's what you're testing for. Just doing the experiment can create the outcome, right? So with the experiment that I told you about that proves the existence of viruses, it can prove the existence of viruses when there's no virus in the experiment. So in other words, it's a false conclusion. The experiment itself produced the results. It had nothing to do with the sample from the patient.
Wow.
That was just coincidental.
Wow, that's amazing. I didn't even know that there were no placebos done on chemo. That makes a lot of sense, but it's like when you see what happened with COVID is just such a classic example where you show someone VAERS, or you use the interface openvaers.com, and you just say, look at this. This is just gives you an idea of how vaccines work in terms of how much injuries they create. Yes, the COVID vaccine did create more injuries than any other vaccine, but by virtue of these systems, but in the reporting system. But everything about the way that things happen, like for example, the way it was marketed, the way it was rushed to market, and sure, the other vaccines took more time, but it was all business development. It wasn't actually efficacy of the product. They weren't testing the groups of people over time, making sure that their outcomes were fine. These never happened.
There was no time, obviously, for any testing. And, this is all because of the emergency status. Everything that was done by this industry, and, you have to say that this was not only the top grossing immunization, but the top grossing drug of all time, right? So these companies, walked away from the past few years, right? Heavily enriched, heavily. But it's the emergency status that allows everything. So governments all over the world declare an emergency. And that means that all of the safeguards that you have to test a new drug to show, like you said, that it works. And by the way, the same thing is true with the diagnostic tests. Not one company that made and sold a diagnostic test for COVID had to actually prove that it was a valid test. In fact, there was no lab anywhere in the world that even attempted to show that the test was valid. So in other words, where they take sick people, give them the test, and show the error rate compared to the sickness. They didn't do that. Nowhere around the world, because they didn't have to. If they tried to do that, they wouldn't have been able to get approval for their tests, because none of the tests are accurate or measure what they're saying that they measure. But the emergency allowed all of these, regulated medical products to simply be sold. They gave them, a carte blanche license to sell the vaccines and the diagnostic tests and make money hand over fist without having to even apply for approval.
Hey, it's Jonathan Otto, and thanks for joining me in episode 2 of my brand new docuseries, Healing Genesis. I just want to say thank you, and that I'm really proud of you for taking the initiative to educate yourself, to make better informed decisions, and to be on equal footing with the elites that try to further their agenda. In this episode, we've learned that COVID is undeniably engineered. It was never a naturally occurring virus that suddenly infected each country and each individual around the world and claimed the lives of billions of persons. Try as they might, there are already thousands of studies pointing to this fact, and the elites could no longer hide this undeniable truth. The scientific papers are finally emerging, and the experts are finally taking a stand and speaking up. We also now know what plasmids are and their role in furthering the agenda of the elite. We are also now well informed that both the government and Big Pharma are undeniably putting synthetic snake venom in us, not only through the COVID shots, but also through the food, water, and even the air that we breathe. It's a dangerous world that we live in, but by equipping ourselves with true healing knowledge and listening to what our bodies are trying to tell us, I firmly believe that nothing, not even the agenda of the elite, can put a stop to our goal of true healing. Thanks again for joining me in this episode, and I hope to see you in the next one.